Embryology of the cranial vault and craniofacial development is a fascinating field that encompasses a wide range of molecular, cellular, and genetic events. Understanding the intricate processes involved in the formation of the skull and facial structures is essential for unraveling the etiology and pathogenesis of craniofacial anomalies. In this script, we will delve into 12 key topics related to cranial vault development, ranging from molecular mechanisms to genetic syndromes and comparative embryology. During embryonic development, the cranial vault is formed through an intricate process involving the differentiation and proliferation of mesenchymal cells. These cells undergo a series of molecular and cellular events that ultimately lead to the deposition of bone and the formation of a rigid cranial vault. Ossification of the cranial vault occurs through two distinct mechanisms: intramembranous ossification and endochondral ossification. Neural crest cells play a crucial role in craniofacial development as they contribute to the formation of various cranial structures such as the bones of the skull, facial cartilage, and connective tissues. The migration of neural crest cells is regulated by a myriad of signaling pathways, including the transforming growth factor beta, TGF-beta, fibroblast growth factor, FGF, and WNT signaling pathways. Perturbations in these signaling cascades can result in craniofacial abnormalities such as craniosynostosis. Cranial sutures are specialized fibrous joints that separate the bones of the skull and allow for growth and flexibility during early postnatal development. The formation and fusion of cranial sutures involve complex interactions between osteoblasts, osteoclasts, and the extracellular matrix. Disruption of suture formation or premature fusion can lead to craniosynostosis, a condition characterized by abnormal skull shape and restricted brain growth. Various genetic syndromes are associated with craniofacial anomalies. Examples include Treacher-Collins syndrome, Apert syndrome, and Pierre-Robin sequence, among others. These syndromes result from mutations in genes involved in craniofacial development, such as the COF1 gene in Treacher-Collins syndrome. Environmental factors, such as teratogens and maternal smoking, can also contribute to the development of craniofacial anomalies. Osteogenesis imperfecta, OI, is a genetic disorder characterized by brittle bones and connective tissue abnormalities due to mutations in collagen genes. Craniofacial manifestations in OI include a triangular-shaped face, prominent forehead, and a small chin. The molecular mechanisms underlying craniofacial abnormalities in OI involve disruptions in collagen synthesis, structure, and function. The skull base provides structural support for the brain and craniofacial structures. Cranial base synchondroses, cartilaginous joints between cranial bones, play a critical role in craniofacial growth and development. Abnormalities in cranial base synchondroses can result in craniofacial growth disorders such as basilar invagination and Chiari malformation. WNT and BMP signaling pathways are crucial regulators of cranial osteogenesis. Canonical WNT signaling promotes osteoblast differentiation and bone formation, while non-canonical WNT signaling controls cranial suture development and fusion. BMP signaling is essential for the proliferation and differentiation of cranial osteoblasts. Dysregulation of these signaling pathways can lead to craniofacial malformations. Neural crest cells give rise to a diverse array of cranial skeletal elements, including the bones of the face, middle ear, and cranial vault. These cells undergo complex migratory patterns and differentiate into specialized cell types such as osteoblasts and chondrocytes, contributing to the formation of distinct craniofacial structures. Suture growth is a tightly regulated process essential for cranial bone development and skull expansion. Molecular and biomechanical factors, including growth factors, extracellular matrix remodeling enzymes, and mechanical forces, influence suture growth. Imbalances in these factors can lead to abnormal suture fusion or cranial growth disorders. Craniofacial dysmorphology encompasses a wide spectrum of abnormalities affecting the structure and function of the skull and facial features. These anomalies can arise due to genetic mutations, environmental factors, or a combination of both. Understanding the underlying genetic and molecular mechanisms is crucial for diagnosis, treatment, and potential prevention. Numerous genetic syndromes are associated with craniofacial malformations, including Down syndrome, Crouzon syndrome, and Treacher-Collins syndrome. Each syndrome is characterized by distinct craniofacial features and is caused by mutations in specific genes involved in craniofacial development. Exploring the underlying mechanisms behind these syndromes provides insights into normal craniofacial development. Comparative embryology allows us to understand the evolutionary changes in skull development among different species. By comparing the craniofacial development of different organisms, we can elucidate conserved and divergent mechanisms shaping the skull's form and function. This knowledge contributes to our understanding of human craniofacial development and evolution. Understanding the development of the skull is essential for medical progress. By studying the intricate processes and genetic factors involved, we gain insights into craniofacial anomalies and potential treatments, benefiting both individuals and our knowledge of human evolution.